Hi everyone, I'm David Del Bourgo. I'm a CEO, co-founder of Whitelab Genomics. We're a company specialized in AI for gene and cell therapy. We founded the company back in 2019 on the premises of developing an AI platform from gene and cell therapy, genomic medicines. We operate from Paris, Boston, and Montreal. We have developed a proprietary platform that uses the advanced AI technologies to help with the application. We are quite agnostic in terms of therapeutic area we address. We work with partners. Our value proposition is we're in silico centric, and we use external wet lab capabilities, or our customers are delivering the in vivo testing or in vitro. We work on both viral and non-viral delivery for DNA and RNA-based therapy. And our specificity is I've developed a guided rational approach based on ligand receptor association. So the whole purpose, as you can imagine, is really to accelerate the development of those amazing therapies that are gene and cell therapy by adding a lot of computational analysis upstream in the process, so you can get to better candidates towards your preclinical phase and advancing faster into the clinical phases. In general, we're not covering everything, although we do a lot of those projects. I just wanted to give a little bird's view on the application of AI. We hear a lot about AI those days. We're happy about it, and where it can be applied in our field. So where we operate actually is really also on the target receptor identification, where we identify receptors that are specific to cells and expressed at the surface. This is where AI can be used. We use a protein language model for that. We also work on vector design optimization. We have very advanced peptide engineering, and we have developed proprietary peptides that are unique in order with binding affinities that are higher than what is seen in the industry to optimize vector delivery. With that, we use machine learning and deep learning approaches, as you can imagine, with protein folding. We also have some projects on payload design, in particular on DNA and promoter optimization using generative AI approaches. And AI, as seen, can also be used to optimize car design and also bioproduction. There is a talk about that tomorrow. We contribute in that field as well, where we help with bioproduction of AAVs and antiviruses. We are like a team of 40 people, composed of scientists mostly, and people also from business development and finance, but we all have a very strong scientific or engineering background, again, based in France and in the US. We're highly computational, so we have a AI tech platform team that's with more fundamental AI, and then protein and vector engineering, but AI-driven. Everything we do is through machine learning approaches or deep learning approaches, including computational biology or just in biology in general. And we have also a part of the team that's taking the AI models to the wet lab, which is external in our case. And we also have a BD team across the world between Paris and Boston. A bit more about the technology. So what we've developed is a proprietary atlas of tissue and cells, where we are able to identify very quickly in different species, different organs, different cell types, surface receptors. So the first thing we do with our platforms, we identify when we want to target an organ, we're going to run our algorithms to identify proteins that are specifically expressed at the surface of a cell and that are specifically to this cell and not to other cells to avoid off-target effects. Then the second part, I can't. Then we have a proprietary database of small molecules and peptides, and we use them as ligands to the receptor. So we have designed in silico and tested in vitro peptides that have like a very high binding affinity, that have specific characteristic that makes them better candidates to be inserted into AAVs, and so then they can be inserted into the AAV to just increase vector specificity. And we also, as I mentioned before, have generative AI project to optimize payload, in particular regulatory gene sequences. As I mentioned, our team, our protein and vector engineering team has leveraged the peptide databases to create our own peptides domain that has proven robustness with increased binding strengths, with a diminution of the peptide size, which helps for the engineering, and the predicted peptide were being experimentally validated. So in blue, Whitelab Genomics peptide domain, and in red, the published peptide domain. We work with partners, as friends from Nanite. We have, though, an internal project where we work on a CNS program where we pushing it ourselves towards validation. The other partnership are with Sanofi, working on ophthalmology, the Vision Institute, also on ophthalmology, not the same disease. With Sanofi again on kidney disease, we have a project with SIREN Biotech, which we probably know, oncology with Nicole Polk. We have also a project on lipid nanoparticles with Debiopharm, and also a project in, again, in oncology with undisclosed biotech, and another undisclosed project on the bioproduction optimization on AAV. A little bit more about what we're doing for Sanofi. For Sanofi, we are using our platform to design better AAV vectors for 2 cell types, for 2 organs, eye and kidney. It's a consortium done with other center of expertise, the Imagine Institute and Nantes University. So as I mentioned, we are targeting eye and kidney. We're designing and generating receptor-specific peptide library to create new AAV vectors to target those 2 cell types, and then to have the vectors validated in vitro and then in vivo. The way we operate is we are R&D partner. So we do pilot project to start a collaboration, but also we enter into larger scale projects where we have more co-development agreement, including shared IP, which is the case in our project with Sanofi. So we are we're a TechBio company, as we say, a technology for biology and not a biotech, but we use some of the biotech models also in our business model. Our platform is named Alfred. In summary, it's quite unique and modular in the field. It also addresses viral and non-viral vectors. So because we have this ligand receptor capacity, we can work on different type of vectors, including other viral vectors or other non-viral vectors than LNPs. We have already traction with major key players in the field, such as Sanofi and other coming in as well. Always happy to discuss new partnership and see how we can help. We're a team of 40 people, pretty agile, pretty quick. And some of the few people who trust us, a lot of people from Genethon, as we started working with Giuseppe. We have also Anne Galli. There is Christian Müller from Sanofi. Professor Bourgo is specialized in peptides in Sherbrooke University. Denise Dalcara. Also, we have a great partnership with the Vision Institute. Thank you for your attention.